right, Dr. Greg Reighard is our next presenter. I'll let you kind of continue along.
Thank you, Sarah. Great to be here again. Of course, after the little freeze last Christmas, we're warming up again. Hopefully, we'll get enough chilling. I'm going to talk about, once again, on the grant that the title is right here, the Solutions to Armillaria Root Rot Affecting U.S. Stone Fruit Industry. You can see there's a number of scientists and others that are involved in it. The ones that are main ones are listed on this slide. This is the areas where the people that are in the multi-state that are working on this. California, though, is primary for almond. The peach industry is not really interested to work with us, but the almonds are working with us. Michigan, it's cherry, primary sour cherry, but all cherry. And of course, in the southeast, you see Georgia and South Carolina, and that's going to be peaches. Kentucky is part of the economic analysis. So I'm going to talk about the peach component in my talk here. And it's not, I missed it. The bottom three are the nurseries working with us, and it's real important. They've been very generous with providing trees and also assistance. Okay. All right, the part I'm working on is breeding-resistant rootstocks. This is an update of what we're talking about this morning. My particular part is the improvement of short and long-term approaches to manage armillaria. Now, the first speaker, Dr. Koch, is part of the short-term part of this grant. I'm more on the long-term. And obviously, our job is to identify resistant germplasm that has the optimal rootstock characteristics for our industry. Okay, I'll just go over quickly. The issues we have in the southeastern U.S. are peach tree shortlife and armillaria. And the armillaria species we're working with is now called desarmillaria, so I don't want to confuse you, but scientifically correct is desarmillaria. Tabescens is the species. Obviously, we want tolerance to ring nematode. That's been our major problem with peach tree shortlife in the past, which is related to the bacterial canker you see in the peach tree shortlife component. What this grant's about is armillaria or desarmillaria. Obviously, that's something that we really have run into a brick wall until recently. Obviously, the rootstock has to be productive and, if possible, vigor control because labor costs are an issue. Okay, just briefly go over peach tree shortlife. This is a fourth leaf orchard, and you see a large section of the trees don't have any leaves. Essentially, what they're doing, they're dying from bacterial canker on this site. They're on level rootstock, and the tree by May has the typical. Well, if you cut it, you'll have sour sap smell, and it's bacterial. What we consider bacterial canker. In this particular planting, there were some guard trees on Guardian. There's 5 trees there. You can see if the leaves. Those trees didn't have any effect, but approximately 80% of the trees on level died. And so this is a research site, and it's known to be very good for screening. I'll get back to that site later because we have a new trial there. Okay, this is what, you've seen some slides on armillaria, desarmillaria root disease, or what we call oak root rot. It fans out from an area where you have infection, and over time will kill any peach tree it gets to. I've done a number of trials where I've looked at over the years for new rootstocks for armillaria and peach tree shortlife. I'm going to talk about all 3 of these sites in this particular presentation. These are the sites where we have the trials. The Musser Farm is more of a upstate Piedmont site that we really don't have severe bacterial canker, and we do have some armillaria, but not extensive, at least on the farm. But Titan Farms has both, has extreme armillaria sites as well as peach tree shortlife sites. Then the Sandhill Station is really ideal for nematode research and also for peach tree shortlife. So it's a very good indicator of resistance. Go first to Titan Farms. This is a trial I could plan 2013. If you look after 6 years, the top 3, Level Guardian and Dihaploid, those are all peach rootstocks, and we had no mortality from armillaria at that time, and neither did we have an MP29. This is in this first column. I guess I got a pointer here. The Prunus sponsoniana, they were small trees. They didn't survive the first year or two. We did replant them, and they did survive after that, and I'll talk a little bit about them. And these 4 down here were releases out of Agrimalora. When I got them, they were Replant Pack and Densiplac, but they've both been trademarked different names. In fact, you'll see Root Pack 20 and Root Pack R in the marketplace here in the U.S. But we look, they started dying after 6 years. A lot of them were dead. By 9 years, they're almost all dead. And we scored them every year for mortality, and the majority of the mortality was due to bacterial canker. Now, the peaches, they started to die, finally, on this armillaria site, and almost essentially all of them that died was due to this armillaria mortality. But after 9 years, and actually, again, this year I checked them, we haven't lost a single tree on MP29. So MP29 is definitely very tolerant, if not resistant, to the species of desarmillaria that we have. This is a picture of the site. You can see how it fans out. You know, these trees were all killed by armillaria. In this same planting, there's a group of 3 trees on MP29. They're all alive. They're reasonably healthy. But they do tend to sucker, especially as the trees get older. They sucker away from the tree. So MP29 is a red leaf rootstock. You can see the suckers in that area, and they can be a problem, but you know they can be handled in the orchard setting. I did replant with Prunus sponsoniana, and they have survived, but they don't. You can see they're a dwarfing. This is on a level rootstock, tree on level rootstock. They do show. Even though these are trees like 5, 6 years old, they do show incompatibility symptoms, so they're not really truly compatible like we'd want. They do produce fruit, and they definitely seem to be very tolerant of armillaria. Another trial in the area, which was supposed to be a severe, it was a severe armillaria trial. Once again, after 5 years, all the control trees guardian were essentially all alive. The Munsoniana, once again, we had issues establishing them. Once again, they were very small trees. But after 11 years, the ones that established, we started losing some, but not to armillaria. We're losing them to peach tree shortlife or bacterial canker. Well, once again, the guardian trees all died from desarmillaria. So it was, once again, it shows, we know peach trees can only last so long on a hot site like that. Once again, Munsoniana didn't show any armillaria death, but they did show death from short life. So, okay, Musser Farm. We have about 240 acres, different sites where we can choose to plant. We're on a peninsula, so we do get cropping every year. For one of the trials, for 30 years, I coordinated the NC140. So all these rootstocks here, we've tested an NC140. The ones you see in yellow, and then the 2 in green, they're the most recent trial from 2017. And the reason I put it on there is because we were able to get MP29 finally in our testing for NC140. So it's in this trial, as well as some root pack. Ones I talked about, that we checked those, wanted to check those, as well as the new ones out of California, which are a controller. This is data from this past year, so it's 6 years in the field, and we're just going to look at the yellow and the MP29 and the purple. MP29 sort of performed what we thought for a vigorous versus a vigorous rootstock like Guardian. MP29 was only about 50% size-wise as far as cross-sectional area, and it was about 2/3 of level. So that's about what we've been seeing. Fruit size has been great on MP29, which we know it does a good job. If we look at yield, the yield is a little bit less compared to the size of the tree, but still was good. The yield was good. But what we found mortality, and it says 90%, but we don't count all the trees. There are 4 tree plots. We only score the middle 2 trees. There's actually more than 90%, or there's actually more than 10% mortality on that planting. This is showing you, we had the reps, and there are 4 tree plots and MP29 right there. But what we found on 2 of our plots, we found this. We had, these are 2 dead trees and 2 dead scaffolds. We only count the 2 inner trees for the data collection. So actually, there's more than 10% mortality. We had 2 plots like that. And so I thought, well, it's unusual. We don't usually get bacterial canker. We get some at the Musser farm, but very little. So I was a little piqued in my interest what was going on. So I got the data from Jackson Springs, North Carolina, and that's that real hot site I showed you earlier. And if you look at MP29 after, this is only after 5 years, there's a lot of mortality. It's only 62% survival, and it's like with the other peach trees. And Guardian was at 95%. And what I was told, the trees on MP29 died from bacterial canker. And Dave Ritchie looked at the trees. Now, he's really an expert on bacterial canker. He took some pictures, and he just showed me the plots. And it's similar like what I saw. You have a dead tree, then you have a tree with a scaffold dead. Another plot, you can see he had the oozing, and he checked it, and he felt it was bacterial canker. In the background, they had Guardian as a border grow, and he hadn't lost any trees on Guardian. So once again, it appears like MP29 can be susceptible to peach tree short life on certain sites. This is an organic peach orchard on MP29 I looked at this summer, and you can see all the red suckers. Most of the trees are dead. There were some trees alive, and when I looked at them, they had the typical flagging and sour sap of bacterial canker. So I don't know. With an organic orchard, it's hard to get enough nitrogen and into the trees. It's probably and it being a semi-dwarfing rootstock, it maybe can't handle low fertility as well. I'm not sure the whole thing, but there wasn't. I mean, it was over 90% of the trees were dead. This is about 5th leaf. So once again, I still like MP29, but I don't know why. Some places it doesn't do as well as I would hope it'd do. Okay, so we know MP29 is semi-dwarfing, and it's armillaria tolerant or even resistant, and we really like those 2 characteristics. But this is at one of the nurseries, and I see the nurseryman's here. But we've tried to do some work with that nurseryman, and sometimes the cuttings, I know we're doing tissue culture now, but when we're doing cuttings, he didn't always get good stands. Then you get this plagotrophic growth where they want to grow sideways because maybe the cutting came from a branch. And then when you have all these flop down liners, you can't really cultivate. It's very difficult to control weeds. And based on some of the growers I've talked to, they were only getting 10 to 20% of the orders when they would ask for MP29. And that's really difficult when you prep for 20 acres and you only get enough for 1 acre. It really messes up your planning. So that was one of the limitations. I know now we can vegetate or we can tissue culture it very well. These are some companies that can do it out west, but we have companies in Florida now doing it. So they can be propagated, at least in quantity. I guess the holdup is getting to a point where they can be budded. So the trees don't cost, I was told $20 just yesterday for them. I mean, it's difficult when you're going to plant 10,000 trees and they cost that much. So this is where we'll get to where, I guess, what we're doing. So we kept looking, even though we have one rootstock out there that works with armillaria, but there are some limitations. We screened a number of different species, all of what we could get material of, and in our screening, 2 species came out with showing some resistance. This is in vitro. Green is the fungus, stained fungus, and red is the root tissue. And for a susceptible rootstock like Guardian, you'll see green all through the cortex of the root system. But 3 selections, 2 are Ceracifera and 1 Munsoniana, they successfully wall out or prevent the fungus from entering into the cortical tissue. And so it looks like, well, maybe we're getting some type of resistance. So when we look in the in vitro, these cultures where we grow the control, then 2 different species of armillaria. We had MP29, then these 3 selections, well, 2 selections. In this case, then Guardian is susceptible. They look going at 2 weeks, everything looks good. But once we get to 8 weeks, MP29 is alive. The control, it doesn't show it red, but it's actually, it doesn't necessarily develop the anthocyanin as well in the study, but in what the system we're growing it. But the two, both MP29 and these 2 selections here still have live shoots, but Guardian, almost everything is dead in the 2 treatments. Of course, this is the control. So it's sort of a quick way, instead of waiting 6-7 years in the field, we're trying to accelerate screening so we don't have to wait that long and we can deal with more numbers. Okay, so we've taken those selections. This is planted in 2021 at Titan Farms on a very severe site. Now there were some, since they're all plums, a little bit of deer browse, and these aren't budded, but the controls are have variety budded on Guardian. It won't go anymore. There we go. This is what they looked like in this May of this year, and I don't know how big they were this fall before they're going to be pruned this winter. But we haven't had a single tree die, so that's good. And obviously we won't see armillaria if it's going to come for another 3 or 4 years. But that was very successful planting. We also planted them at Sandhill Station because that's where we already have severe peach tree short life. These are some of the former trials there, or 4 tree plots. You know, it's sandy, but it really weeds out susceptible rootstocks. Here's a 4 tree plot of one of the European stalks. They all died in the 2nd or actually 3rd leaf. So it's a very good test site. These are some of the trials I've done in the past there where we put this trial on, and in all of them, Guardian held up very well. Guardian and even Level held up very well. In this case, we didn't have either. We only had Helford as a control, and this was a later planting. All the Helford died very quickly, and the Munsoniana we put in there was holding up fairly well. So I guess the bottom line is it's a very good site to test these rootstocks, not so much for armillaria, but we'll make sure that they can tolerate bacterial canker, peach tree short life. This is the crew, and they're on their planting. This particular planting has deer issues, also has weed, or has a lot of issues. The first year didn't do so well, so we'll do more intensive management of it on this past year, and this is what it was this past spring. So I think they're going to make it now and until Short Life decides to take them out. But it's a very good site for screening for peach tree short life. So, no observations from screening rootstock cultivars and selections. I've been doing it for a long time. Really, no peach seedlings have survived better than Level or Guardian on, as far as any type of site we've planted, except if it's oak root rot. Obviously, they're both susceptible to desert armillaria. MP29 has been superior, or you can call it a resistant rootstock on severe oak root rot site that we tested, and obviously it's done well in other rootstock or severe oak root rot sites in the southeast. So I think it's proven itself. It has shown, at least in the studies that I've done, every place where I've had bacterial canker and even where I haven't, I'm seeing bacterial canker on it, and I don't know why. I mean, the material is correct. And of course, it's been difficult to propagate sufficient trees to meet the demand, and of course, everyone's still working on that. The rootstocks from Europe, like replant Pack R, is supposed to be tolerant to armillaria in Europe. It dies very quickly from bacterial canker in the southeast. And the others that they're pushing for intensive systems, I've tested them at 3 different locations in our state. I've also had them tested in the NC 140, and any place where you have bacterial canker, they have not done well, in the southeast for sure. And the new selections, the hybrids that Dr. Gasic is making a lot of crosses. We're trying to incorporate what we want. And there's a lot of molecular work that I'm not talking about, that where we've isolated haplotypes and gene sections that seem to have the type of genes that seem to be correlated with or attached to certain rootstocks that do well with armillaria and for peach tree short life. So we're, through her breeding program and using molecular genetics, we're hoping to get a lot of new things to test, and obviously then we need to evaluate them for field resistance, not just in the lab. So, and lastly, anything we produce has to be easy for the nurserymen to propagate. And if they can't do a good job on it, then we're back to where we are with MP29, really having difficulty producing the numbers. So with that, this is, acknowledgments to all the people. And of course, I'm working with Titan Farms, added them on there. So, thank you. Do we have time?
You got time? Anybody got questions?
Yes, sure, Monty.
So for the trees that were affected with peach short life, what was the population density of them?
What was the what density?
Population density, like how
Population
many
density.
nematodes?
Oh, the nematodes. Actually, like the site at Mustard, we should have been low. We pre-plant fumigated. In fact, also the trial at Jackson Springs was pre-plant fumigated with talone. So I didn't take any samples after that because at Mustard we have, we have ring nematode, but it's mostly ornata, which is on Bermuda grass. It doesn't attack peach. Xenoplex is the one we worry about, but I've never seen high numbers in my orchards at Mustard, but we still fumigate. Now, Jackson Springs, I don't know if they took nematode samples later, but they did fumigate, pre-plant fumigate there. But as far as the history, we say if we get more than 50 per 150 cc, then you have too many. And they use, by the third year, they get to that level in areas where you have short life. We always see it. The fumigation only works for 2 years. You got to get your trees up, because after that, the nematode will be coming back. Yes, Walker.
How are we coming on mapping the genes for them? We have the peach genome and with regards to root rot and short.
Well, Senya should answer that. We have some very interesting stuff, but till it's published, I don't think we're going to give it out. Senya.
Yeah, I mean, we have this session kind of joint and stuff. So I'll talk in the microphone if it's recorded, so it's recorded there. We are not doing mapping because that requires developing mapping population, and then that takes time. What we are trying to do is to understand which genes are active during the tests in vitro that Dr. Reighard showed, and we found some that confirmed in other species with other fungal pathogens. So it's the same group of genes, and so we know them, and we are now developing the tests so we can faster identify the material that has those genes and then move forward. As Dr. Reighard said, I'm not going to say which genes, and because the paper is in the process, and once it gets published, then it's going to be a public knowledge. But right now, we are just keeping everything under the wraps. And what I also wanted to say, we did not include it here, all of these selections are given to Cumberland Valley Nursery to bud them and to test them and to tell us do they like them or how do they perform, because we are really conscious about issues that could be down the stream. We can develop anything that's great, but if nurseries cannot produce the trees, then there's no value in it. And so I'm just curious to see how it's going to perform, because last year is when we gave them the plants to bud, and so it's going to be a continuation story. Also, one of the additions for the socioeconomic analysis and all of these, the paper we published for the RCE effect on armillaria and the orchard longevity was published in 2018, if I'm not mistaken. So we are continuing collecting the data. We have like 3-4 more years in addition to that to see how many years we can prolong the life of the orchards and how many trees live longer than normally they would if there was no RCE. And that's probably we need to talk to Guido to maybe publish some short note to that study now that we have more years. Because as you know, we are trying to get results out as soon as we can, but with armillaria in particular, if you have a field study, you have to wait 5, 7, 8, 9, 10 years to get valid data to report.
We're getting there, Walker, but it takes a while. I remember starting molecular genetics around when Abbott was hired 1989. And at that time, we promised in 10 years we would know everything. Well, we didn't get the peach sequence to what, 2008 it was released or whatever. So it takes a lot longer sometimes, but there's been a lot of things happening. And we're not even talking about gene editing. We have a colleague that's doing gene editing with this situation, and he's had some success too. So we're making progress, but it's unfortunately for the growers, it might be we hope before your children take over the business, but it's not easy. So.
Any other questions? We have a couple more minutes.
I'd just like to add, my experience with armillaria in South Carolina is I don't really see it, and my data shows, I don't really see it till 6th or 7th year. But Dr. Schnabel says he sees it 4th year or 3rd or 4th year. So, yeah, so I think the growers I see, they do a good job of root raking, get as much out of the orchard as you can. And even in the most severe sites, I don't see it till the 6th or 7th leaf. And I think that's partly due to the effort of the growers to get everything out of the orchard when they replace it. Phil, what about those trees? You said some of them budded pretty well?
We were real impressed with a couple of those varieties that they sent us. Of course, we don't know how compatible.
It'll take more than a year. I mean, some of that's delayed in compatibility. Also,
At this point, we were impressed with them, but this is just the beginning.
yeah, you got 10 more years, then you can say that. So. Well, thank you.
All right, thank you both for that. And then,
Yeah.
are you doing it?
Yeah, I mean, both of us can just
Okay,
cover.
Dr. Gasic and Dr. Vassalos are both going to go over the armillaria survey.
I'll just start, yeah. So initially, when we were planning this session, the idea was to actually conduct the survey right now. That's why there's so much time for this particular group of talk. But then we decided that it's much more user-friendly to say it if county agents approach the growers and talk to you and conduct the survey. So what we wanted to just now emphasize, and then I'll let Dr. Vassalos even go into the scientific side of it, how important is for us and for you to participate in this survey, because what we tell you after the survey depends on how many farms participate and how significant those farms for production are that participate in the survey. And that's why we get sometimes the data that don't make sense to you, because maybe some of the key farms did not participate in the survey. And I know, I personally hate when I get emails with the survey, I just don't even look at them. And so we think it's much better if you have a friendly face with the county agents coming to you, explaining what the survey is about and what you're going to get out of it once we get the data processed, and then conducts it on spot with you. And that's what we are doing, not just for the peach, we're doing the same approach with the cherry and with almond in California, just to make sure. And also, we're all sensitive about sharing the contact information and blast emails and all sorts of things. So county agents have their email lists, and they can either contact you via email or directly. So please, if you get that email, pay attention to it, because we really depend on your collaboration for this part. And so, Michael.
Thank you, Dr. Gasic. So like Dr. Gasic said, it's so important for us and for you, right, to get reliable results on the impact of armillaria and even to provide some data on nurseries on what the farmers want. We like to have information from you using this survey. Oops. Now, I know, right, that you get tons of surveys every day on your mailbox, on email. We spent a lot of time with focus groups, even with that phone survey we did. So we can promise you that the survey will not take more than 20 minutes of your time when you met with the extension agent. You will be in and out, the agent will be in and out of your house or your farm within 20 minutes, and we will have all this information that will be confidential. No one will know who did the survey, but we will have the results that we could publish. We can inform the nurseries. We can even give some information to you about the impact of armillaria. Now, the survey, the questionnaire you will see with the extension agents has 5 sections. The first part of the survey talks a little bit general information about your farm. The second part and the third focuses on armillaria. So on the second part, you will have questions like the one I presented from the phone survey that basically talk about how many trees, if you had armillaria on the farm, how many trees died, how much money you spent approximately to treat armillaria, what type of approach you follow, if you do root collar excavation, if you do root raking, if you use chemicals. All of this is very interesting, very important for us. The third question will just give you some examples of potential rootstocks and the price per tree. And we would like from you to tell us if, for example, a new rootstock is developed that there is an improvement in resistance in armillaria by 10 or 20%, how much more you are willing to pay for that tree. Because this will help the nurseries also know, right? Okay, if we can develop that, but it's costly, will the farmers buy it? That's what we want to get out from you with that third question. The fourth type of question, it's just a couple of general agree, disagree questions on how you market your produce. Where do you see the future of the industry in terms of marketing? So do you think it will go more direct marketing? Do you think it will have more contracts with the industry? Do you see more CSAs happening? So we would like that information, and then we end up with typical demographic questions that help also USDA and help all of us know who is actually the peach farmer. So again, it will not take more than 20 minutes of your time, and we would like to have as many of you as possible complete the surveys, because it will give us a lot of information that will help the breeders, that will help the nurseries, and at the end of the day will help all of you. Let me know if you have any questions about the survey.
Also, what I forgot to mention, this way we will not bombard you with the numerous emails to complete the survey, because the county agents will have the list of people that did it, and so you do it once and you're done. So that's also one of the benefits. And what I wanted before we go into the questions, if you have questions, and I want to thank you all for your support, because without your support this would not be a possible endeavor. And we are doing all the best we could, but you are the ones that provide us the input on what's important, how to direct the research and where to go with it. So this is just not just for this project that I'm involved in, but for the general that peach team at Clemson University does the work. It's all directed to help you and to support your productions and organizations. And then we really are appreciative of all of your help and participation in our work.
When do you think the agents will go out? About a month from now?
I'm not sure that's...
We have plenty of time. If anybody has any questions about this or about any of the previous talks from this morning's sessions, feel free to ask them.
the survey?
Rural area in general or the survey? Oh, the survey.
We want South Carolina, Georgia, Florida, North Carolina. Potentially, we had some from California, but mainly the Southeast. One more thing, if you're way too busy, right, because we respect your time a lot, you can tell the agent, and we can send you a link or a QR code so you can complete the survey online, and it's still anonymous. So if you think that this will work better for you, we can give you a link.
Yeah, right.
This is not just peach, okay? So,
Yeah.
cherry, you definitely want Michigan people, and some Michigan people do grow peaches up there, keep that in mind. And for California, it's probably the almond board, and I'm not sure if any of the peach growers, some peach growers grow almonds and peaches, so. And Colorado, well, Colorado doesn't have armillaria, but I thought we were gonna, you just want the Southeast and then California.
California, yeah.
Talking about peach, though.
Correct.
Yes,
Peach,
okay,
yeah,
just-
yeah, yeah.
just-
Okay, yes, I'd be clear about that.
Mhm.
If you're from Mississippi or Alabama, yeah, you're welcome.
So, any questions? Thank you very much for your attention. Yeah, go ahead, Greg.
Some growers are very large, they have multiple, people involved in organization. You just went from one person from each organization.
Correct. We want the person who makes the decisions for each organization.
Some organizations have multiple people who make decisions. Different decisions.
Oh, correct. So we want the one who makes mostly the decisions about rootstocks used, what.
So you want whoever makes decisions in the field versus who makes decisions in the packing house?
Correct. The field.
It's so complicated, but they're gonna like who's supposed to fill it out.
We will correct. We will like the person that makes the field decision.
But that's why the county agents are involved, because we realize that there's much more complexity in just sending an email. Who answers? Then you can get multiple answers from the same farm because we don't track who did answer because it's anonymous, and so we'll do the best we can. But some of these demographics questions and some like a general type of question will serve to direct the analysis, so we can actually say, okay, this is the size of farms that answered, or this is the region and stuff like that.
But once again, get back to my question. Yeah, some people manage, what plants, where they're going to plant what. But there's someone who makes a decision on how much replacement for their operation, what percent of the orchards are they going to replace each year. So I think it's going to be more than one person that needs to talk together when they answer this on some.
On the big farms.
operations.
Yeah, correct.
Don't actually, we only want to talk to one person, but actually, there might be two people or something.
So as an agent, I guess our jobs can be I will reach out to each farm and just include everybody in that. And potentially the best thing is just to sit down with, let that farm make the decision, send it out to everybody and know, maybe let's just sit down and have a meeting. That way we can kind of do a little round table to make sure that we're gathering all of the information to make sure that we have, all the I's dotted and T's crossed.
In one of the slides, you showed that the rootstock showed the resistant one created a barrier between the armillaria and the tree. So was any molecular studies done to find out what was the composition for something, what's the barrier?
Yes, we did not include the general slides about the project. So the talks today refer to the objective 3, which is the horticultural aspect of the new material that we find in the field towards the resistance for these diseases, and even for the nursery aspect of the quality of those rootstocks. Root collar excavation is the short-term help to the farms to sustain their production until we can come up with the rootstock that will cover all of the issues. Right now we have MP29, but obviously the more research and more fields that it was planted, some other issues pop up like with everything. Nothing is always foolproof. We have objective 1, which we use basically to find the new sources of tolerance. Then we test them in that quick test that Dr. Reighard showed in the in vitro, because we don't have a life to wait 7-8 years to see it in the field. And then once we test and find out something that's tolerant, we submit it to the root microscopy to see if there is a penetration of fungus, or it's just a fluke that plant escaped the fungus in the in vitro. And once we get their confirmation, then we work on the protocol to develop the in vitro propagation method. So that's out of the way, so we have no issues propagating it if it ends up being something that's a good rootstock. And then when we propagate material, we give them to Dr. Reighard to plant them on a hot site for peach tree short life and for armillaria. We give them to nurseries, Cumberland Valley Nursery, to graft them and to tell us we don't like it, it's not good, and now we have the grafted rootstock. We don't still know how the take will be, and if there maybe later on will be some incompatibilities or some issues. Time will tell. And so that's first and third objective. And the second one covers your question. We basically look into the genome, transcriptome, and metabolome. And also we are looking into the under the histological analysis of the tolerant material to see not just metabolically what they do, but do they build the barriers. And so we have a paper in the pipeline, I think, and one is already published about the type of barriers the tolerant material builds to wall off the fungus. So sometimes it penetrates, but the plant is able to sense it and build the wall and stop it. So all of it is covered under the project. Today was just about something that directly can be helpful for growers, because they don't have 10, 15, or 20 years to wait. They need help right now.
You still are making crosses too.
Of course, yeah. So anything that we find that's tolerant, we are making crosses, and then it goes into the pipeline to screen for the tolerance, and that's basically Alejandro sits here. He's the postdoc that slaves off on those parts, and so Dr. Guido Schnabel is our pathologist that works with us to do the testing. So all of the sites are covered. Yeah. Yeah.
I came in late, I missed the presentation, I apologize, but we're supposed to know the genome of peaches and plums, right?
Yes.
Do we have any markers for armillaria resistance?
Working on it. We sequenced the...
We don't have the markers.
Not yet. We sequenced the genomes of the MP29, of Guardian, because the peach genome, when you say peach genome, it's a really like a floating message. Yes, we have a peach genome sequence, and published in 2013, I think was the first publication. But that's the genome of the haploid, or the half of the genome of the Lowell rootstock. So, because of it's a haploid and diploidized, basically, so we have everything homogeneous, and that made it such a good genome sequence. But that's half information only for Lowell. So when you use that to develop the markers, you're basically missing the information that's in the new material that you don't see. Yeah, go ahead.
The Guardian rootstock's been out a long time.
Yes.
Y'all select some things so that it would germinate better for the nursery, right?
Yes.
Over time, right?
Yes.
So how the genome of that rootstock, how many variabilities, how much variability is in that rootstock?
That's a good question. We did not actually, because that would require sequencing the genomes of all of the variants we have, and Dr. Reighard has more than 100 trees from that initial population. But what we did it different way. We collected the seeds and we tried to develop the in vitro propagation protocol for Guardian, and we've seen that there are variability among the seeds, and so from those we are trying to select the ones that would best propagate. But we did sequence the original plant, that's the Guardian rootstock that we are using right now, and we are using it in the molecular studies. Going back to your question about the...
Then, the Guardian rootstock's been around a long time. And my question is, what do we know about it as far as resistance and the markers to resist nematode? Do we know exactly what those markers are for that?
We did not yet develop the markers. We are testing them. We have a few because we do know where the region for the tolerance is on the chromosome 2 and like a top part of the chromosome 2, and that's general tolerance to the nematodes. But the markers was not developed because for that you need a particular segregation progeny, and that took time, and the material that was used to find that region was not available anymore. So we used a different approach, and now we have the region and we are testing it, but it takes time. So right now, unfortunately, we were stopped in tracks. Yes, I know. This year is the goal. Both. We already are developing markers for malaria, and within this project, the funding is there to do both, and so it's going to go in parallel. So I'm hoping by the summertime we will have, or by the springtime when we have the fresh leaves in the field to collect and to do the testing, we could start testing the markers that we developed. Yes. Yes, but there was no, we operate on competitive funding from the USDA, so the rate of funding is not that great. And so, when we get the money, we do as much as we can. So we were lucky that, unfortunately for you guys, that armillaria became such a big problem, so we actually got the funding for it, and that moved the needle on the research. So, Until
we get some markers, we really don't, we're just kind of selecting on a phenotype situation, and it might not be so diverse, it might not be quite as bulletproof as we think.
You're correct, but also markers are great, but they can just direct you and make you more efficient in what you're doing. I'm a breeder and I use them a lot for certain other things, but we don't put like all our hope on them. So what we actually developed within this program is the quick method to use within 6 months. We can test the material in vitro and show if they are tolerant or not. So now what we're going to do, we're going to use the markers and go back. So just a bit of a backup story, how we found these few selections that we are now testing in the field. We collected everything from the Prunus collection in Davis, California. They hold there all relatives, wild relatives of peach. We collected, we tested them, and then we found those few in vitro that they are tolerant to armillaria. Then we confirmed with the root microscopy and now in the field.
One more question. Can you inoculate tests in the greenhouse with armillaria tests?
Not in the greenhouse.
You can't even do it.
No, armillaria doesn't like.
That's really the book where we go. You can't, in the laboratory, go down the line and really scientifically, it's hard.
We can in vitro.
But not in the greenhouse.
Not in the greenhouse, yeah. But in vitro is much faster, cleaner, and we can control better than the greenhouse. The problem with armillaria is because it doesn't grow above the soil line, and that's why the berms work, because it doesn't like the changing environment. It likes the stable of what's in the soil, and you cannot recreate that in the greenhouse, because those pots, I mean, if it's hot, if it's cold, there are changes, and armillaria doesn't like that. And I mean, if Guido was here, unfortunately, he got succumbed to COVID. If he was here, he would tell you it's not for the lack of research. There's been 10, 15 years, people are trying to develop the method, but it's not consistent. And so I cannot repeat it and get the same results as you, and then scientifically you cannot use it.
We have, as far as bacterial canker, the San Jose site will screen them out within 3 years. By 4th week, they're dead. So that part, even if we don't have a marker at that point, that will screen for Peachtree shoreline. That's how we got Guardian so quickly. We screened 700 genotypes, it was the only one that survived. And it's held up for 28 years. But for the armillaria, what we're doing is the best we can, and it seems to work.
Yes. Yes. I mean, the time is our enemy. You know, the grant is 4 years long. We planted those trees, so hopefully we will get you, request to you for another 4 years when we go to apply for the funding. So all of these trees that Dr. Reighard showed you that are planted, we can continue evaluating them because we need time. And so Dr. Schnabel also showed that if you plant on the hot spot where basically orchard is not, or the land you cannot use anymore for replanting because of armillaria, then you can see the effects in the 3rd or 4th year. But that, again, doesn't mean that we can see it within this grant, because it's only 4 years. So whatever we planted now, fingers crossed we get funding for another 4 years, then within that project we'll actually hit more ground in a sense that we'll go into the fields, we'll evaluate, we'll look and plant all of the material and work more closely with nurseries as well to just make sure that if something shows up and stands to the test on the Peachtree shortlife and armillaria in the field, that nurseries are already ready to propagate it, that there's no lag on that side.
I think if you look at what we've looked at, MP29 is resistant, okay? And in these containers we're growing it, these others are performing, showing the same type of resistance. So I think they probably are resistant. But we know MP29, at least for what we have in the southeast, is I'd say almost resistant to armillaria. So even though it may take 6 years out in the field, where I'm already feel good because it took a number of years to get this to grow them in these containers of media that they'll grow, and these selections that we made, they survive just like MP29. And when we do the histological sections and stain them, same type of thing. They wall out the fungus. So I'm hopeful, very hopeful that we'll hopefully confirmed. It might take a while, but right now I think they're equal, probably MP29 in that regard, but maybe not the Peachtree shortlife. So like I said, we're trying to get something with both things.
And also we don't know the effect of these new materials as a rootstocks on the scion for the production-wise, size of the fruit or all of the other things. That's why we need time. But luckily now that we have the Cumberland Valley Nursery budding, I'm not going to say producing the trees, we'll see how successful that's going to be. And if there are trees, we'll take those trees, we'll plant them, establish the orchards with a scion, and then the rest will continue. So within next 4 or 5 years, we'll have a field data to convince you what we see already in vitro about the tolerance, and to actually add to that how those new materials as a rootstock affect the production, because that's ultimately the problem as well, that we have to know the answer to. Yeah.
Where are we at with regards to tissue culture and system analogous to stem cells in animals?
Oh, that's a tough question. So you mean in relationship to the Armillaria project, or so? Not sure I will have an answer for you right now. But what we are doing, and for all of these. Ah, okay, okay. Sorry, then I misunderstood. Peach. Okay, so peach is. Easy in some things, but tough in some. And one of the tough things is to regenerate it from a single cell. And we have some promising research done within our group that I'm not allowed to talk to, but hopefully very soon that is going to be published, that we found a way to propagate, to clonally propagate it from a single cell. If you do some modifications, so being it transformation, being it any other way, that was part of the problem with the peach. You could not regenerate it from single cell, and the peach as an organism, if there are a mixture of cells, which we call chimeras, and there are wild type or normal type cells, they will overpower the others. So within 5-6 months in the greenhouse, you basically have a wild type peach. Right now, we found a way, we found a system to do the somatic embryogenesis, which basically means from a single cell, you add a, what would be the word? Create the embryo.
It does work, and you can't publish it until,
Yeah, yeah.
But this has been a problem for 30 years in peach.
Even more.
I think Dr. Sasky, I guess, Dr.
Yes.
Sasky, I think, he took a platform from cotton he developed, tweaked it for peach, and it's very impressive, the data, so.
And that was funding outside of this grant. So that's why I'm not allowed to talk about the results, but they are very promising, and we are hoping that once it's publicly announced through the publications, then we can actually use that system for all of the genes that we determine that are active with Armillaria, peach tree shortlife, or any other things, that we can just do the engineering of the guardian if we want a guardian, or if we want some other cultivar, because it wouldn't be the transgenic plant, it would basically be modifying those genes to be expressed more or shutting them down if they are resistant genes. But that's another talk. But what we are trying to do on the tissue culture side for these rootstock, what's actually really good is we already have the tissue culture system to give it to any company that's massively producing the rootstocks, that they can run with it. We have the media conditions, everything for these that Dr. Reighard showed in his presentation. So we are able to clonally propagate them with just the facility and manpower to do the work. Okay, I think we filled the time nicely. Thank you very much for questions. If there are more questions, we'll be around and you can ask us.
Oh. Yep. Thank you, everybody. Thank you for asking your questions. And thank you, Dr. Gasic and Dr. Reighard, Dr. Vassalos, for answering those.